This is a production of Cornell University.
Okay. Awesome. It's really great to be presenting back in this room. The last time I gave a seminar here was my job talk many, many years ago. So this is very exciting to be back giving my tenure seminar. Before I jump into the science, I've learned to overcome. Okay, before I jump into the science, I really have to thank my amazing lab members, both past and present, who are just an incredibly strong and active group of people. I flipped back through my photo reels all the way to 2018 when we started and realized that we moved lab spaces 3 different times so far, hopefully one more time when the renovation's finished. And there was almost no complaining during that time. Just an incredibly adaptive group that continued to science through the pandemic. My first grad student graduated kind of in the midst of the pandemic and was just incredibly productive in that challenging time. And now as we enter another challenging time, I'm just really proud of this group for always looking for ways to push the science forward. So most of my lab's work focuses on plant grafting, although as you heard, we had some soybean money, so we've done some other stuff too that I won't talk about today. I'll just focus on the grafting. And grafting is simply the physical joining of separate plant parts so that they share an interconnected vascular system. And throughout this talk, I'll really emphasize that second part of the definition, the interconnected vascular system. That's really crucial to our understanding of what makes grafting work. And grafting's pretty remarkable. It's our way of making real live Frankenstein's monsters. And it happens incredibly easily in the plant kingdom. So grafting actually occurs in nature. These are 2 examples of kissing trees that we saw in Yosemite on a hike. And this is a self-fusing tree by the Ithaca Farmers Market that you can see here. And historians think that it was human observations of these natural grafts that inspired the first agricultural applications of grafting, which date back in the literature to 424 BCE, and it was likely co-invented in ancient Greece and in Asia at the same time. So when I say old tools for maximizing agriculture, to some extent, these are prehistoric tools and really anciently historic tools in human hands. There are many, many applications of grafting. I'm just going to point out a few prominent ones that we might care about. So grafting is used across viticulture to protect vines from biotic stresses. There's a very famous decimation of European vineyards from the phylloxera outbreak that was in which the vineyards were saved through grafting to Missouri rootstocks, wild Missouri rootstocks. And so if you enjoy wine, you might appreciate the benefits of grafting in this case. Grafting is also used to modify plant architecture. So here's an example of dwarfing rootstocks that make apple harvest more accessible. This is also used in pear harvest. In the reverse direction, in vegetable crop production, grafting can be used to invigorate the shoot system or scion and greatly improve yields in tomatoes and cucurbits predominantly. And that's a direct topic of my lab. Grafting is also used in horticultural art. This is an example of 40 different varieties grafted onto one stalk. And probably one of the most recent innovations in grafting is that you can harvest ketchup and fries from the same plant. And you can actually buy this in the UK. And it's covered on a Stephen Colbert episode, so it's very famous. Grafting has wide horticultural uses, but in our case, most of us probably read about grafting when we're studying mobile signaling. And so it's also used to demonstrate the movement and signaling function of peptides, small RNAs. An emerging field is messenger RNAs that we're doing a lot of work in, and hormones. And so grafting is really the gold standard for demonstrating mobile signaling. And that was this area of grafting was really the extent of my knowledge when I entered into the field of grafting as a postdoc. And then as I learned about its prominence in agricultural production, I really became fascinated with the concept that we could actually use modern molecular tools to understand some of the molecular biology that underlies beneficial agricultural grafting and potentially start to engineer beneficial grafts for agricultural systems, which I think we're really at the cusp of doing just now, almost 7 years into having the lab. So we do most of our work in tomato and the broader nightshade family because it's very easy to graft. I won't show you this video just so I have enough time to get through everything, but look it up. It's produced by a Cornell alum, Dr. Molly Edwards, who is also in Mike Scanlon's lab when I was a PhD student, and she has a whole series of science IRL videos. So it's very easy to graft tomato. It has almost 100% success in grafting, and it heals quite quickly relative to woody crop systems. And so this is the shoot system or scion and the rootstock, and this is the joining graft system, the graft junction that's formed 10 days post-grafting. And so we have a very popular technique that we use in the lab with confocal imaging of cleared tissue, where we can actually image into that graft junction and study the vascular connectivity that occurs as the two parts of the plant heal together. And so here we're getting into the vascular system, and you can see that the regenerative processes that are occurring give this beautiful stitched together xylem and phloem system. And so we can look at how that is disrupted and the genes that are involved in placing that vasculature so the plant can reconnect, partially using this anatomical imaging. Another reason to study grafting in tomatoes is that they are widely grafted for commercial production. So how many people in the room knew that the tomatoes that you buy at Wegmans or your favorite grocery store are grafted? Yeah, because I've been here a few years, but still not everyone. So pretty much all tomatoes produced through controlled indoor agricultural systems are grafted to rootstocks that both invigorate the production of the scion as well as provide abiotic and biotic stress protection. And this is a trip that I took the very first summer of starting my lab to the Netherlands, where I visited a plant propagation facility where they were getting ready to graft 100,000 plants to ship to the Middle East for production. So it's used around the world in agricultural production. And so hopefully some of the stuff that we learn can influence some of the practices in positive ways in tomato production. So despite the fact that grafting has been used internationally for millennia, there are still some core questions that motivate the exciting biology that we do in the lab. And so one really exciting and challenging question is how different graft combinations produce emergent phenotypes, especially phenotypes that are beneficial to us. And then another question is just what moves across the graft junction and to what extent are mobile molecules functioning as signals? And we're really focusing on mobile messenger RNAs here, which I don't have time to talk about, but I will point you to some great resources from my lab during the talk. And then finally, at the most basic level, we don't have a good understanding of what determines whether or not two species are going to be compatible with each other, whether or not they can form interconnected vasculature during grafting. And by compatible, I mean the ability for two things to coexist without problems or conflict. So the ability for them to stitch together. And so we study our questions in this compatibility framework where two things can graft, but maybe they have like no appreciable beneficial impact on each other. On the other end of the spectrum, two things that are incompatible have a problematic coexistence. So they're unable to coexist in that graft format. And then on the beneficial side, ultra compatibility is where two things can graft and that graft produces beneficial outputs that we appreciate as humans. So not evolutionarily beneficial, but appreciated by us through our lens. And so a lot of the work that we've done is on this spectrum of compatibility and incompatibility, because my first student to join the lab, Dr. Hannah Thomas, was super excited about these questions. So she really carved out a lot of work in this direction that Elise Boisvert, PhD candidate, and Sam Yanders, a PhD student, are now carrying forward. And so graft incompatibility is not a huge problem for tomato. Tomato grafts pretty well to its wild relatives, but it can be a big problem in woody crops, and it can go undetected for long periods of time. And this is really a classic graft incompatibility between pear and quince, where you can see this necrotic divide across the junction. And by detecting that incompatibility, growers can then graft in an interstock that is mutually compatible with the rootstock and the scion, which is super cool. So you can actually rescue these incompatibilities if you can detect them. We think that the information that we learn from tomato is going to provide useful markers for detecting incompatibility in these other systems, because the process of grafting is very similar across different species. So when you graft two things together, there's an initial adhesion that is that occurs through the release of pectins that help stick the cells together. And then a wound response process promotes the formation of callus. And so that's like an undifferentiated wound responsive tissue that then starts to make contact, and the callus will start to form plasmodesmatal connections and communicate across the graft. That callus fills in, and through that space, vascular differentiation and other tissue differentiation starts to occur. And then subsequent the resumption of physiological transport and subsequent differentiation leads to a very, very strong junction that is actually stronger than any other part in the stem, because you now have extra vasculature, extra liquefaction. And so this whole process involves local signaling to actually place the vascular strands in the right place to reconnect the two plant parts. And that process is not understood at the molecular level. The use of vascular patterning and differentiation genes, and all of these processes affect the integrity and biophysical strength of the plant at the end. And you'll see how that informs some of the tests we use to look at compatibility. So to understand compatibility in our system, we grafted tomato to a bunch of different nightshade relatives that were readily available, and just looked at whether or not there were negative impacts on the plant in response to grafting. And we found a really pronounced negative impact on pepper-tomato combinations. And so this is self-grafted tomato, and this is tomepper and pepmato. And I think this picture really accentuates the impact of the negative graft. This is tomato on a cayenne rootstock, grafted at the same time as self-grafted tomato. And so often these plants are physiologically impaired, which leads to stunted growth. And so it's a pretty clear incompatibility phenotype. But importantly, the plants do live, otherwise it'd be hard to study them. And so we consider this system an example of delayed incompatibility, where they're unable to graft, but they're able to survive for long periods of time. And I've actually had a tomepper plant live in my office for over a year. So they can live and grow very, very slowly for a very long time. And so a lot of Hannah's work looked at what causes this incompatibility in pepmato. And before I dive into it, I'll just show you some of the detailed information we have about what happens when tomato is self-grafted. So when we have a compatible graft forming. And this was collected in collaboration with nuclear physicist at WashU Med School, Dr. Yuan Chuan Tai, who just had a unique passion for plants and built his PET imager, which allows the tracking of radio-labeled nucleides through 3D space and time. He built it inside of a growth chamber, so you could do physiologically relevant experiments in plants. And so we use that to track a labeled carbon CO2 movement in grafted plants that were sealed. They had an airtight seal right above the junction. What you can see is that at 5 days post-grafting, we start seeing the resumption of carbon transport. And so that indicates that physiological transport through the phloem happens as early as 5 days post-grafting in this setup. And then we used labeled nitrogen to track xylem transport because labeled water has a very short half-life. And we saw that the plants resume transport as early as 6 days post-grafting. And so within less than a week, you have a healed junction that's transporting. In PET tomato, we didn't have access to the PET imager, although I'm pretty excited about the new XLEAP proposal. I think the synchrotron will have similar capacities in a few years. So we used a fluorescent dye called CFDA, that's flow immobile. You can see the aphids that are contaminating our plant are taking it up in their bellies. One example of good contamination, I guess. And when we look at CFDA, it gets stopped at the junction. And so we looked closer into the anatomy of these grafted plants. And what you can see is in self-grafted plants, you have these nice long spaghetti strings of xylem running through. And then in PET tomato, we have this disorganized vasculature. So it's forming these anastomoses and failing to connect and communicate across the junction. We see a very similar phenomenon in the reciprocal direction. This is now with forming O staining for lignin. And you get this mass of differentiating vasculature that's failing to communicate and coordinate with the other half of the graft. So we consider these bridges to nowhere. And we actually have a bridge to nowhere here in Ithaca. How many people know the plans for the bridge to nowhere in Ithaca? Okay, so it's pretty exciting. It's getting connected to a major trail system, so it will become a bridge to somewhere very soon. And that's kind of our hope with this work too, is that one day we can rescue these incompatibilities, but we're not there yet. And so basically the plant is forming bridges, but they're not connecting across. They're not reconnecting the physiological transport. And this has major consequences for the biophysical stability of the stem. And so this is a bend test that Hannah invented. And basically you bend the plant, and if it breaks at the junction, it fails the test because that junction is supposed to be the strongest point in the stem. And if it breaks anywhere else, it passes the test. And so Tomepper and PET tomato almost always break at the stem. Every once in a while you get an exception because it's biology, but almost always break at the stem. So another thing we looked at was whether we could identify some genetic regulators that are controlling this compatibility. And so we collected RNA-seq data of PET tomato and Tomepper grafts and tomato and pepper self-grafts at 1, 3, and 5 days post-grafting. And we worked with Dr. Rastuzani's lab at NC State to build what I think are like above average pretty gene networks. They look organized to me at least. And they were able to identify putative major regulators of graft formation at the 1, 3, and 5 day time points, as well as mixed time points. And then this is for self-grafted tomato. The networks for incompatible grafts were crazy and really hard to navigate. It was like this huge mess of dysregulation, I guess. And so what we did was we mapped on the loss of connectivity in the network to look for genes that were important in compatible graft formation that are disrupted in the incompatible grafts. And this is just an example of one gene that we see that has altered connectivity. It's PTI5 that Dr. Greg Martin's lab happened to have mutants for because it interacts with AVR-PTO, one of his favorite mutants. And so this is cool. This was implicated to function in grafting at 1 day post-grafting, which is when those cells should be adhering to each other. And so we see that the vasculature connects. The graft actually forms okay, but there's this fissure that remains at the wound site. And we have more work to do to understand what's going on there, but it's kind of a neat example of the networks informing some of the biology. So one thing that surprised us was that most of the hubs in the self-graph networks and in these incompatible graphs are not related to vascular patterning, but are big, most apparent phenotype in these incompatible graphs is disorganized vasculature. And we think that that's because a minority of the cells in the harvested tissue are actually forming new vasculature. So it's a bulk RNA-seq data set with a lot of different things happening. And so they're less pronounced in the network. So we pulled out known vascular patterning genes based on a network from Arabidopsis that's really nicely characterized. And we built our own network from these genes based on what's differentially expressed across our time points and graph types. And so in this network, there's a lot of hypotheses to be tested, but one was the role of this transcription factor called LOX4 that's involved in cambial specification. And so one indication that it might be important in this process is that it has somewhat chaotic expression in Twimeper and Pepmato relative to self-grafted, where it has this gradual elevation in expression. And we are also able to obtain mutants for this from Zach Lippman's lab, which is very nice. This was in 2020 when it was challenging to do lots of wet lab work. And so you can see these WOX4 mutants. They're relatively normal. They're a little bit stunted in growth, but generally they look like a normal tomato. They make vascular bundles, so they have a nice stem anatomy. And when we graft them, they have high survival rates. And so this is self-grafted wild type. This is self-grafted WOX4, 100% survival. It was pretty disappointing. But then Hannah performed the bend test and saw that 100% of them actually break at the junction. And so that's an indication that there's a loss of biophysical stability, even though the plants survive. And so that's like maybe our big PR message from the lab is that survival doesn't equal compatibility. We can actually look at the anatomy of these WOX4 plants relative to wild type that form vascular connections. And we see that they are impaired in specifying vascular connections. And Elise is looking more into the extent to which this occurs. So it's pretty cool that these networks helped us find, one regulator in the process of vascular specification. And if we go back to the vascular network from Arabidopsis, you can see that there's other potential genes that are likely involved in placing the vasculature in the right place so that reconnections happen. So WOX4 is the one that we looked at, but upstream of WOX4 are these small signaling peptides called TDIFs or CLEs, and CLE41 is one of those genes that help specify WOX4 in the process of vascular patterning. And so we're also able to get a mutant for that. And it looks like it's disrupted again in the vascular specification. And again, Elise is looking into the extent to which this occurs and whether this really alluring signal could be playing the major role in placing vasculature in the right place so that roots and shoots can communicate post-grafting. And so one question that we've asked ourselves after collecting all this data and looking at these mutants is whether we've really explained graft incompatibility. So does WOX4 look like our Pepmato and Twimeper mutants? And I'd say not really. So in Pepmato, we have disorganized vascular proliferation, and in WOX4, we just fail to specify the vasculature. So these networks are telling us about genes that are essential in grafting, but maybe not explaining the phenomenon of incompatibility. So Hannah dug deeper into the role of tissue death in incompatibility. Again, this is in 2020, it was dark times. And so she just looked at basically the persistence of dead tissue at the graft over long periods of time using this trichem blue staining, where this deep purple color means that the cells are dead, and these lighter purple colors are kind of just noise in the imaging. And so she looked at 7, 14, and 21 days after grafting. And what she found, she quantified it using an ImageJ pipeline. And what she found was that there's similar levels of dead tissue 1 week after grafting in self-grafted plants and incompatible grafts. But over time, the self-grafts will clear that dead tissue, and the incompatible grafts persist in having dead tissue. So of course, you wanted to see what was happening in terms of gene expression in this persistent tissue death process. So she did RNA-seq at these time points as well. And this time she pulled apart the scion half of the graph and stock half of the graph. So this top half is scion data, this bottom half is stock. And she just looked at differential expression and saw that the incompatible graphs, this is aligned to the tomato genome, and this is the pepper genome. A lot of factors here. The incompatible graphs have a dramatic upregulation and downregulation of differentially expressed genes. There's a lot of Go categories that are enriched across these time points, but there are some that point us towards some really cool models for what's happening. So one, which is just a nice reality check, is that there's a response to water deprivation. It's not making xylem connections, so that felt right. It's still unclear how plants live for this long without making xylem connections. That's an open physiological mystery. But in addition, there's a lot of these gene ontology categories related to the plant immune system and defense. And so Hannah's big hypothesis from this data, and she did a lot more digging that I won't go into, but her big hypothesis is that the incompatible combinations of genomes are causing this activation of the plant immune system that's leading to cell death. And if you want to hear more about kind of our approach to digging into the molecular details and mechanisms that might be working within this this hypothesis, go to Sam Yender's seminar next month, the student seminar. So another question that we looked at is whether pepmato and tomeper are a specific type of incompatibility, or whether there are some general rules that guide incompatibility in this branch of the Solanaceae. And so what Hannah did was she did these reciprocal grafts across a few members of the Solanoideae that were possible to grow and graft together. And we ignored Nicotiana and N-benth tobacco because there's some like pretty wild reports of Nicotiana being able to graft like across pretty much any flowering plant family. So it's like a super grafter that's doing something exceptional. So we stuck to this branch of the Solanoideae. And it doesn't look like a ton of plants to graft together, but once you start doing the reciprocal grafts in replication, it's actually a huge experiment. And again, all the grafts survive. So survivorship is not a measure of compatibility. Self-grafts form really nice vascular connections, string through the junction. Intrageneric grafts within solanums, this is tomato and eggplant, form beautiful vascular connections. And this is work done by Michelle Heaney, a PhD candidate in the lab. And in fact, this system is so strong that it's formed a system that she's using to look at long distance communication between species. But all intergeneric, so across genera, grafts fail to communicate and form these really, really ugly disorganized grafts. And so we did a ton of confocal imaging to look at this process, but we also performed the Bend test, and that's a pretty good indicator of compatibility. So pretty much all the incompatible grafts here break almost 100% of the time with the Bend test. So you really don't have to do imaging if you want like a fast measure of this, this property. And so to kind of wrap up this part of the talk, we have found many ways to make grafts fail. And also collected some really great tools to study this in more depth. So we've really expanded the tool system here. And again, Tom plant and Igmato is another system for studying communication, and Michelle was just awarded the best 3-minute thesis in Cal, so you can watch her talk on YouTube or hear her talk in a real seminar for much more in-depth view of the process. But we're hoping to use these tools to eventually find ways to reconnect those bridges, so make graphs succeed. And that, I think, will be a tremendous effort to overcome incompatibilities, but it's going to be exciting. So I want to use the last part of the talk to talk about this other end of the spectrum, where we're looking at compatibility, this neutral coexistence, which is really home plant and egg mato. They don't really have huge beneficial impacts on each other, and ultra compatibility. And so this is an area of work that's really kind of brand new results coming out of the lab. And this is looking at the ability to program communication within plants for ultra compatibility from the viewpoint of the farmer or the human. And so the goal here is to actually reprogram the way plants are communicating to give us more information about the environmental context in which plants are growing, and enhance the timing and quantitative responsive management practices from our end. And so the first kind of example of this reprogramming that we're looking at involves the signaling system called the CEP signaling system, C-terminally encoded peptide. Say the name says it all. And this is a root to shoot and shoot to root signaling system that allows roots to forage in heterogeneous nitrogen conditions. And it's partially responsible for kind of the classic high nitrogen, low nitrogen split plate response that's described in the literature, where roots will branch and grow more extensively in areas of high nitrogen. And so how this works is that under low nitrogen, roots will start to express these small peptides called CEPs that move from root to shoot, where they're perceived by a receptor called CEPR1, which turns on the production of a downstream signal called CEP downstream, really great naming scheme here, which moves back down from the shoot to the root and influences nitrogen foraging responses, including upregulation of nitrogen uptake transporters, as well as increased root branching in places where there's high nitrogen and not where there's low nitrogen. And so our idea was that we could actually plug into the signaling system to learn about when plants are experiencing below ground nitrogen stress by seeing the output of this below ground signal in the shoot, and then actually reprogram the activation of the signaling system to turn on in response to other below ground stresses. And so this is a huge collaborative project, and these are the major postdocs and grad students who have been leading this work across my lab, Sijin Lee's lab, Abe Strook's lab, and Mark Bausen's lab. And so what we've done is we've created these reporter plants that tell us when CEP downstream is turned on. So that's telling us when CEPs have moved and turned on the response by turning on a Ruby reporter. So this is a pigmentation reporter in tomato plants under low nitrogen. You can see it's really nicely expressed in the phloem, the flanking phloem region, where it's then going to produce a downstream signal that moves back from shoot to root. And we've also made fluorescent lines in Arabidopsis to look at a more quantitative response of this process. And so this is the exciting kind of new data coming out of the lab where the plants actually work. And so up here is wild type tomato, and down here are these Ruby lines under 13 days of nitrogen deprivation. And so a grad student in Abe's lab, Jacob Belding, has actually quantified using a really simple image processing pipeline that he developed, the response of these Ruby lines under low nitrogen over time versus wild type plants. And the cool thing here is that you actually start seeing a significant response within days of low nitrogen treatment which is way before any grower is going to see that with the yellowing of leaves and the lost productivity of their plants. So we think we have this early alarm system to help people manage their plants that can then be adapted to many different below ground inputs that are hard to manage. And so we've also built this in Arabidopsis with fluorescent reporters where you see the kinetics are quite fast. So within 6 hours of low nitrogen treatment, we see this reporter turning on. And I thought this is quite a rapid response. And now the big question is, when does it actually turn off? Like, so if you apply nitrogen, can you turn the signal back off and use it throughout the season? And so we'll be getting into these messier questions in real field environments as well as in the lab. So we have plants that are actually started already in Tucson, Arizona, and they're going to go out to Maricopa. And then another set of plants that are going to be here in Aurora, New York, growing amongst cornfields. So this is really, really cool to see work from our lab going out into the real world to be tested. Well, the quote unquote real world, the research environment, but still the field to be tested. And then again, the next step is really to see to what extent can this communication be reprogrammed to tell us about other below ground events that are really hard to monitor, like drought and pathogens. Okay, I think I have 4 minutes. So I'll tell you about, I'll end with actually the first grafting phenomenon that got me into grafting. And this is the phenomenon of grafting-induced vigor, where when you graft to a hybrid rootstock, an interspecific hybrid rootstock, you get this boost in vegetative and reproductive output that increases yield. And so the commercial rootstocks are typically formed through these crosses between domesticated tomato and a green-fruited tomato called Solanum haberchides. And one of the most popular commercial rootstocks that we use in our research as well is Maxifort, or Max for short. And so when you graft to Maxifort, the domesticated tomato shoot system has this huge boost in its growth. And so here's just the increase in biomass that you get when you graft to Maxifort that then influences an increase in yield. And that's a large reason for why the grafting is used in tomato production, in addition to abiotic and biotic stress tolerance. And so a really cool thing with this phenomenon is that it's highly repeatable across environments. So these are greenhouse, growth chamber, and multiple field studies showing that the Maxifort rootstock really increases vegetative biomass and yield across environments, which is not common in biology to find something that repeats across environments. So one of the questions that I actually started out asking is whether it's really the hybrid state of the root system that's increasing the biomass of the shoot. And so what I did is I compared yield in a domesticated tomato grafted to a haberchides rootstock versus a hybrid, an F1 hybrid that I made with this haberchides crossed to M82. And I compared that to the influence of Maxifort rootstocks. And so what I saw was that it's really the hybrid state that boosts yield, but also my random hybrid that I made just because the seeds were available in the lab performs pretty similarly to one of the top performing commercial lines. And that's because I think we're growing these in field conditions, and a lot of rootstock breeding is targeted to controlled agricultural environments. And so the very first grant in the lab that we got was a Hatch grant to look at whether we can identify other rootstocks that are beneficial for outdoor New York State growing environments. And so we made 30 different hybrid rootstocks with different haberchides ecotypes and grew these in high tunnels in Freeville. So this is a very popular way to grow tomatoes near here. And pretty much any farmer's market tomato that's available earlier in the season or later in the season is likely harvested from one of these high tunnels. And we found that many of our experimental rootstocks marked in green perform equally well to some top commercial rootstocks, or sometimes even a little bit better. And so this is another example of us going, really out into the real world, where we were able to take our top performing rootstocks, test them out at a major production site 2 hours from here. So Zaid Kurdik welcomed us and our crazy tomatoes. And we didn't get a quantitative measure of whether or not they liked them, but this is a quote. Wow, they're significantly taller and healthier than the non-grafted plants in the middle. And so Tian is working on a more quantitative measure of, what influences whether or not people use grafting in their farms. And so she's performing a real intensive survey at these grower expos, which has been really fun to go to. And so just like how the Hebrides probably has untapped potential for positively influencing production and the ability for plants to perform, Nora is looking at how other wild tomatoes are also influencing the performance of tomatoes. And so she's making a ton of other hybrid crosses to these different, some of these different, not all of these, but some of these different wild species that have superior stress capacity, and see whether you can like rapidly adapt tomato to challenging environments. So that's, I think, a really cool direction to work. So to wrap up, we have a ton of awesome tools for the spectrum of compatibility to incompatibility and ultra compatibility. And of course, more questions than we started with. And I have to thank the lab again for carving out these questions and leading the work forward. Our collaborators and people who really generously provided resources. I don't typically do this, but because it's a tenure seminar, my mentors, especially Adrian Roeder, the head of my mentorship committee, who's made tons and tons of time for me during the last many years. And then funding, without which none of this would happen. So hopefully funding continues. And that's it. Yeah, I'll take questions.
Is there a relationship between whether you can recover hybrid F1 hybrids easily and graft compatibility? Because I know like Pinelli and Hebrides there and the kind of that tail end where you don't need to do certain like embryo rescue and that kind of stuff to get hybrids.
Right, yeah. So is reproductive compatibility an indicator for graft compatibility? And no, it's not. So there's a really cool study where a group in Germany actually overcame reproductive barriers between two Nicotiana species through grafting. So they were able to fuse the genomes of the nuclei basically into one new allopolyploid individual that could set seed. So like the graft compatibility is much, much broader than reproductive compatibility. It's what that showed us, at least. It's really cool. Yeah.
How was Max originally selected, given that Hebrides seems to be almost as good? Is there like
other Hebrides? I think that there's a suite of other traits that are probably highly relevant to controlled agricultural environments, including probably stacking of disease traits, in addition to its ability to promote that grafting induced vigor phenotype, because it is one of the most invigorating rootstocks that we've evaluated, and we've looked at many. So it's definitely like a surefire way to increase vegetative and reproductive biomass, which can be a problem. Sometimes people have to it takes a lot more maintenance to keep the plants in check.
The question about the incompatibility that involvement of immune response. I guess I will hear more from them. But I was just thinking that is it immune response, suppression of immune response? Is it a continued process or it's happening at the very beginning? Once it overcome, then it's mainly the developmental process that involves.
Yeah, that's such a great question. And I think that's right on Sam's mind, is like which part's active in allowing plants to graft, and whether it's like an active process to be able to graft or an active rejection of incompatible grafts. So we don't know, but we'll know in a couple of years.
So I don't really know anything about plant grafting, but in fungi, there can be some interspecific fusion, but like there are lots of strains of the same species that don't talk to each other, and that has really helped in finding these like immune response mechanisms. And I'm wondering if there are like in plants, if like there are different isolates of like tomato that maybe are in the same species or separate species, but like don't talk to each other at all, or don't pass the Venn test or whatever, that you could maybe use to help get at some of that immune response questions.
Yeah, that's a great question. We were thinking about maybe integration lines where you have just chunks of like a really distant species that could help with mapping the genetics of it. But tomato grafts pretty well to like the wild relatives that we've tested so far. So, but there are examples in cucurbitaceae, so cucurbits, where there's more distinct incompatibilities within a species. So we should look at and try get adventurous. Yeah.
How adaptable do you think the nitrogen reporter plant could be to like remote sensing via UAVs?
We're gonna test it out. Yeah, so there's a drone that will like fly over the Maricopa fields and the Aurora fields. We'll see. I'm like a detailed phenotyper, so I'm a little skeptical, but the field people aren't, so hopefully I'm not. Yeah.
Are there any parallels to this to like parasitic plants?
Yeah, definitely. They're
A useful tool to explore.
Yeah, so that's helped us kind of hone in on some the next set of candidates that might be interesting in those networks. So there's these LBD. There's a lot of these lateral boundary domain genes, but there are ones that are essential for host parasite interactions in tomato that are also expressed in our data set. So they're like high level candidates. Yeah.
Great. Yeah, thank you.
This has been a production of Cornell University, on the web at cornell.edu.